Yeah, I would do like physics.
Okay, so we'll go ahead and get started. A couple announcements. Module 4 exam, we'll start that Wednesday. I have it mainly written. I have to write all the answers now, and I'm dreading that. So you'll have that Wednesday for 24 hours. Again, Module 4. If you notice, the syllabus as uploaded in Canvas is a little ambiguous about Module 5. I was originally going to have it sort of synthesis-based, but I think we're just going to do the biomolecules of life. So we've already covered carbohydrates. We'll revisit them a little bit when we get to DNA. But maybe I can take an informal poll. In terms of doing something different, I try to mix it up each semester. I was thinking rather than having a Module 5 exam, we would just have activities that are going to be based on these bracelets you make. So the point of the bracelet, other than arts and crafts, now that you know small molecules, we're going to start talking about large molecules, and they can be difficult to conceptualize. And so as I'll explain on the next slide what the purpose of the bracelet is, we'll carry it through to the end of the semester, and then you're going to use that to do some stuff. And then you're going to make another bracelet, which is going to have a different sequence in it to mimic a short strand of a biomolecule of life. I don't have everything uploaded into Canvas just yet in terms of tracking these activities, but I was thinking since your exams are worth 60 points, I don't want it to make it overly cumbersome on you or especially me in terms of grading. So maybe like 4 or 5 activities related to these bracelets, and then you will not have a Module 5 exam. So if you just do the basics of the activity, you'll get your points. I'm not out to scrutinize. As you'll see with these things, it would take me a lot of time to say, okay, this is exactly right. But as long as you understand the logic, hopefully you'll see the point of the activity as a replacement for the exam. So is everyone, let's do it this way. Are there any objections to that? Seeing none, we'll move on. I'm sitting on Faculty Senate this semester, and they use Robert Rules of Order. If you're not familiar with that, maybe check it out. And all I can say is it's amazing that the U.S. has a Declaration of Independence and a Constitution, considering once you get more than 2 people in a room, having them agree on anything is amazing, I think. So I used to do this as like a bonus, but now I'm just going to sort of do it as an activity because I think the learning part of it will make the concepts more real in a sense. So I'm using a term out of med school here. We'll call this a practical. And so you got a piece of twine. You have your 10 colored beads. You just picked them based on whatever color. Unbeknownst to you, those colors are going to represent an amino acid. And then stick them on there however you want. And then you're going to upload a picture of that into Canvas. And then I'm going to have a key of what those colors are in terms of the amino acids. And then you have to tell me the sequence. So we're going to learn about, we'll review what the acids are, then we're going to see how do you make them in organic chemistry. And then as what we have, including this one, I think we have just 5 lectures left. Don't everyone cry at once, but so the amino acid is going to represent, or the bracelet's going to represent a small peptide. So peptides are made by how DNA is read. And so from that bracelet, the second bracelet you're going to make are the codons from which those amino acids go in that sequence. And so that's how we'll learn about peptides or enzymes, which are amino acids, and then RNA and DNA, which are nucleic acids. And so the sequences of these chains impact the function on which they do things biologically. And so today we'll just run through the amino acids, things that are important to them, for you moving on to other classes as well, and then we'll end on sort of the big picture of this, why I think it's sort of cool, even though I'm a straight organic chemist. I think hopefully you'll find it interesting as well, as humans have thought about why we exist here in this place and time. So just watch for Canvas assignments for these activities, but each will probably be 12 or 15 points apiece. I'll set the due dates the last day of the semester, but if you got it done, just upload it and make your own life easier. So we already went through these first couple slides about the amino acids when we were talking about amines, but we'll review them, and then the next part of this is because it's an amine and an acid, you have these sort of orthogonal functional groups, an acid and a base. How do they behave under different pHs? And that affects ultimately how in the lab these things would be purified. So as we talked about previously, we're dealing with really the alpha amino acids. So we know the alpha carbon is the sp3 hybridized carbon next to the sp2 hybridized carbon of the acid. And so there are 20 of these, and if you counted, there's 20 colors there. So each color represents proteinogenic amino acids, and the naturally occurring amino acids have the S configuration. So when you look at the Newman projection of this, or excuse me, the Fisher projection, you're putting your I there, and that means the amine ends up on the left, and so don't confuse this, but the other nomenclature, this is the L. So S amino acids have the L configuration. So remember that naturally occurring sugars were D sugars, where the OH was on the right. There's 4 classes. So nonpolar, polar, acidic, and basic. And those classes relate to, since the acid and the amine are common, they relate to the side chain, which I've highlighted in blue there. So nonpolar are typically just aliphatic, so sp3 hybridized carbons and H bonds, nothing else. Well, I don't want to say nothing else because some of these have heteroatoms. Polar, you have another like OH group or an amine group or some other heteroatom functional group that gives it polarity. Acidic and basic just have an extra acid or basic functional group in them. If I was sitting down for an exam, I could write the structures of these after teaching it for so long now. But if you do move forward in biology or biochemistry, you'll want to know, what valine is, just in your memory, so that, because you see it so often, okay, it's nonpolar, and that will ultimately help you as you progress in your studies. But those are the words. From the words, there are 3-letter abbreviations. You can kind of make out what they are based on how the letters are cherry-picked from the name. And then what you'll see in the key for your bracelet is just the one letter. So from the 3-letter, there's the one letter. And for a lack of a better way of saying it, those amino acids in those one-letter designations are an alphabet of which larger molecules will be created. And again, I haven't fully fleshed this out, so bear with me and don't crucify me on my evaluations, which I've been reminded to tell you to fill out. So the Nobel Prize this year in chemistry was rewarded for this software called AlphaFold, which is associated with Google. And so what it does is, in silico, in a computer, it will computationally predict what the active conformation of an enzyme or protein is. So I still have to look and see if you'll be able to enter your sequence in there and have it model it. But that might be one of the activities I was thinking of. So just to review, here's your name, the 3-letter, and the 1-letter. Yeah, I mean, if we look, maybe we can conservatively say most of them just start with the letter. So glycine is gly or G. But if you look at tryptophan, TRP, that's W. So things don't always translate exactly. But the top row, that side chain is all sort of hydrocarbon-like, so those are nonpolar. The bottom row, so methionine has S-methyl. Notice it's not SH, it's S-methyl, so that makes it a little bit nonpolar. Proline is the only one that's cyclic. So the side chain is tied back to the amine group, and it's also the only one that is a secondary amine. Phenylalanine has an aromatic ring in it, and then tryptophan this is called the indole ring. So the unique thing about proline, as we'll see, is since it's a ring, when we learn about protein structure, that ring is going to help move the next amino acid in a certain way. So it's going to limit the conformational mobility. And ultimately, since enzymes essentially react non-covalently, you have something come in and then it leaves. The structure of the enzyme is determined by the sequence of the amino acids and then ultimately its conformation. So as we see the polar amino acids, asparagine, now we have an amide out here. Glutamine, you're just adding a CH2 spacer, but it's another amide. Serine, you have a primary alcohol. Threonine, now you have a secondary alcohol. The other thing to notice is this has another stereocenter in it as well. Tyrosine. So tyrosine looks like phenylalanine, except now you have this, which is the aromatic alcohol called phenol. And then cysteine looks like serine, except for OH you have SH. The acidic ones, as I said, have the second carboxylic acid off the chain, so aspartic acid and glutamic acid, and then the difference between them is the linker. The basic ones, arginine, this group is called a guanidine. Histidine, we talked about the imidazole ring. And then lysine is just a floppy chain and then another amine, so that's a primary amine. So essentially those are the building blocks of peptides and proteins. So the next thing that we'll look at, if you look at the structure here, you have an acid. And then you have this amine. And so another way to think about these is they exist in a Zwitter ionic form, where like the amine base has deprotonated the acid. And so why do they exist in that form? Because the pK of the ammonium group, it'll depend, but it's about 10. So remember, the acid base always goes to the weaker pair. So a Zwitter ion is also called an internal salt. So you have the plus and the minus. So what we'll look at is when you're taking the individual amino acids and changing the pH of the solution, how does that change the plus and minus charges that you see of that amino acid? And then ultimately, when you get a sequence of them, like multiple amino acids, like in your chain, what's the overall charge of that sequence? So like right now, this has 0 charge. But as we change the pH, the charge will change. So we're going to look at pHs from 0 to 14. And as we see, 5 and 10 lie within 0 and 14, so those charge protonation states will change. So as a refresher, here's our pH scale. On the low end, from 0 to 7, that's acidic. On the high end, from 7 to 14, that's basic. And then for just ease, 7, which is neutral, you can think of as physiological pH. So it says, for structural considerations, the acid has a pKa of 2. So that means to deprotonate that. And the amine we're going to think of as that. So I said 9, 10, it's around there. So when you're within 0 to 14 pH, that correlates directly to pKa. And so if we think of the overall charge, if something's more positive or if it's more negative, when you stick it in a medium with an electric field, you can move that towards the cathode or the anode, and I never remember what is what. But basically, if you're moving it, think of it like thin layer chromatography, where instead of moving things based on functional group polarity, you're now moving them based on charge. And if you're able to do that, that's a separation technique. So that's called a gel electrophoresis. Has anyone ever done that? So I've never done it. How big are the gels?
You can get really small ones, or you can get like really big ones. It just depends on what you're trying to do with it. When I did it, I was in like middle school.
Middle school?
They were really little. It was a middle class.
Okay.
I just worked this on a few days ago, and ours are typically like
So maybe like 10 by 10 centimeters or something.
But
We
we've been
have
shooting.
some in our lab that are like this big, but for like really big things, you can get like a foot,
Yeah.
but it's also very expensive because you have to like make a medium, so you don't want to do like the big ones.
Yeah.
Try to get it done about that big.
And so let me understand if I'm teaching this, but obviously I'm ignorant of it. You load it from the top, right? And then stuff moves down.
Yeah. He has the
Well, it has like wells that you make, so you can, it's like
Yeah.
almost a comb you put in there, and you have to get through the well, but not into the bottom of to the plastic.
all the way
It's-
down.
You don't want to disturb it, basically, is that? So, but then these things are migrating because there's this electric charge
Mhm.
differential, right? And then when it migrates, do you see anything?
I think I have it.
It's like very thin line sometimes.
So you
What I did, I saw it was like thin lines. We were doing something with DNA, and you could see where.
You see a
certain messed up.
band?
Mhm.
So how do you visualize the bands if you don't see them?
There's a little, that or like a UV thing, like a little chamber, you put it in and take a picture with it.
Okay, so like a UV light then. Okay. So it's the sort of biochemist TLC, if that's fair to say. And now these questions that we do, just think of it in terms of that, how these things are going to migrate in terms of how you would separate charged molecules. So due to the nature of that R1 side chain, for the nonpolar, they'll all sort of be the same because you don't have these other acidic or basic functional groups present. They'll vary in their charges, and that's how you get the migratory ability. So the charge will have these different charge states. And again, when it's 0, that's called the Zwitter ion. So let's imagine, in the middle here, since you have the amine plus the carboxylate minus, the overall charge is 0. Plus and minus, that gives you 0. And so that's at a neutral pH of 7. So that's our reference point. But now let's say we're starting at pH 0. So I like to think of these one way, and then as we're moving through it, we can go back and forth. If you're at a pH 0, that pH is less than 2.34. That's the pK of that acid. So that means because it's acidic, the amine has to be protonated. So you get the ammonium. The carboxyl is also protonated. So you can think of it as +1 plus 0 equals +1. So that's where this number comes from. Now, if we're titrating in base, that means the pH is going up. You're eventually going to deprotonate the acid after you hit 2.34. That means the base deprotonates this proton. So now you get a negative charge at the oxygen. So plus and then minus, that gets you the 0. And as you keep adding in more base, you're going to deprotonate the ammonium to give the amine. And now overall here you have a negative charge. And so what, do you typically keep the pH constant when you do this? What,
You put a buffer in there, hopefully.
what is the buffer region, acidic or basic or?
I think you try to keep it neutral, but it also depends on like the materials you're using, like the safety data sheet, all that kind of stuff.
Okay.
Specific.
Yeah, buffers is where I hit a wall. I'm sure a lot of students do. Because the buffer region, again, that's basically you want to keep the pH constant based on the stuff you're adding. You don't want it to spill over because that's going to change the outcome. So this is a a organic chemist way of thinking of a titration curve, you could then plot this of charge versus pH. And because we're dealing with charge, there's this new term here called the isoelectric point, or PI. And that's defined as the concentration in which the concentration of the Zwitter ion is the greatest. And so really what you're doing is PI is looking at the two pKa values. So we'll say acid and then ammonium, and then divide that by 2. So for alanine, you end up with a PI of 6. So at pH 6, the primary form is the Zwitter ion, then there's no charge. And so again, if you wanted to give it a charge, you would adjust the pH to move it either to the negative or the positive region. So that this is the table for the nonpolar amino acids. And really, if you look at the first or the second two columns, pKa1 and pKa2, the values are pretty much the same, right? Because the side chain really isn't affecting any protonation states. So overall, you're around 6. So once you shove those two numbers in, divide by 2, that gets you the fourth column. And if you look at them, they're pretty much 6. So we'll do another one, and I'm going to cheat. Since I don't have the answers in front of me anymore, I'm going to make sure that I don't spread misinformation. And I'm going to look at my prior slides. So proline has the first pKa at 1.99, the second pKa at 10.6, and then the PI therefore is 6.3. So the question states, maybe I'll diagram it here. So 7, 6.3. There's 14. If you were at a pH of 13, what would the form of proline be? So I guess another one I could put in is the 10.6. What?
Is it B?
Yeah, so it's going to be B. So again, if you're thinking from left to right, and the pH is already at 13, you're under basic conditions. So at that point, the acid has been deprotonated, and now because you're beyond 10.6, the ammonium has been deprotonated. So the ammonium must be neutral, the acid must be negative. So a pH of 4, so 7, 6.3, 0. So now we're at 4, and the pK1 was 1.99. So now we're in acidic conditions, but 4 is greater than 1.99, right? So what do we think the structure here is? So yeah, this one will be C. So since we're acidic, the amine is still protonated, but we haven't reached the pH yet that will protonate the carboxyl. So the polar ones, I guess for those 6, what I've done is separated them out into two categories. The first of which does not have the extra OH or SH. Or wait, yes they do. Serine has OH. Okay, so the second table they're separated out because that third functional group lies within 0 to 14. The first table, even though serine has OH, the pKa of that OH is around 16, so that lies outside the region. But the take-home message for these, maybe you're around like 5.5 now as the average. So cysteine, the side chain is SH, so that's more acidic than a normal alcohol. And then tyrosine, you have the phenol, so that essentially if you look at the pKa3, that's telling you where that gets protonated or deprotonated. So cysteine is lower 5, tyrosine falls within the other ones 5.5. So there are the three for tyrosine, 2.2, 9.11, and 10.07. So the PI overall is 5.56, and if we're at a pH of 11, what would the charge state of this one be? So since 11 is beyond all of them, the answer would be D. So we can think of this one as, I'm going to say that one is 10.07. I'll say this one is the 9.11, and then this one was the 2.2. So to reiterate, it's the chemical reaction is an acid-base reaction, and then you're sticking it in that pH scale to see where the pH is versus the pKa of protonating or deprotonating. So overall, this would be a -2 charge. So we've 14, 7, 9.11, 10.07, and now we're at a pH of 9.5. So I might have screwed up the numbers on the last slide, or the associations on the last slide. Based on the structures that we see here. So at that pH, the carboxyl is definitely going to be in the negative charge. And so based on what we see in terms of the possible responses, I think this one has to be pKa2, and that one has to be pKa3. So I might have mixed those up on the prior slide. Since now at 9.5 we're below 10.7, the ammonium has been deprotonated to form the amine, while the phenol is still protonated. So here we get a negative 1 charge. So now we're at pH 4. So we're in the acidic region. So at this point, we've sort of surpassed 10.07, 9.11. So those functional groups must be protonated. Does that sound logical? And so we haven't hit the 2.2 yet, which belongs to there. So that one still must be deprotonated. And then if we look at what's left, plus and minus, this would be the charge of 0, so that's the Zwitter ion. So we get B, and now, since there's only 4 questions, 4 responses, so this one's A, because the pH now at 1 is below 2.2, so that means the acid has to be protonated. So we're at an overall charge of +1. So since we're moving that way, past that pKa, the carboxylic acid is protonated. So I think we have 2 more examples of this. We'll go through the acidic and then the basic using the same logic. So aspartic acid and glutamic acid, notice pKa1 is due to acid 1. That's typically, I believe, the one closest to the amine. Then pKa2 is your other acid, and then pKa3 is of your amine or ammonium. So notice these pi values are much lower, approximately around 3. So we'll start at 0, draw the line to 7. It says the pH is at 2.5. And we have the first pK1 at 1.88. The second pKa2 is at 3.65. So at this pH, the amine should definitely be protonated, right? So I think we can eliminate D. And then one of the acids is going to be deprotonated. The other one should be protonated because we're in the area between the two. So that would eliminate A. And then I think we can eliminate C because both of them are deprotonated. So this should be B. And that is charge of 0, so that's the Zwitter ion. So now for the basic amino acids, pKa1 is the acid, and then you have pKas 2 and 3 are the different amine groups. And so you'll see these have a big range, right? So there's really not an average number that we can target for this. So let's draw a 7. PK1 is 1.82. PKA2 is at 6. And then PKA3, 9.17. And so we're concerned about what is the structure at pH 7. So at this point, the acid should be deprotonated. So that would eliminate A and B. And so since we're past 6, one of those amines has to be deprotonated. So just using the logic of between C and D, that would eliminate C as well to give D as the correct answer. And so since that charge is 0, that's the Zwitter ion. So just to highlight that, pKa2 was the protonated imidazole. So it's clear that, some of you have definitely more practical experience doing this in lab. I've never done it. And so you probably have a better understanding of what is actually happening than I do, which is good. So if I do talk out of turn, feel free to correct me. So I'm educated. So again, gel electrophoresis, it's a solid phase gel that's pH buffered, and that electric current is applied through it so that you're migrating these things to separate them based on charge. And so when you're doing one of these, do you cut the band out and then analyze that, or is that not part of normal?
We have a printer, like the thing that I was talking about that takes the picture, just like prints it directly onto a piece of paper, and then you can use like a key for different peptides. Say if you're wanting to like see what this proteins are in a specific molecule, you just put them next to each other and kind of like do the cross reference, like, oh, this is probably, this peptide, that kind
Yeah.
of stuff.
So there's never, but can you cut that gel out, or it's that you can't work with it after?
When you cut the gel out, you have to cut it out for a specific reason, as with extract it
Yeah.
or something with it next.
Yeah, to like further analyze it. So you have like a reference band, and then you're horizontally moving over to see, does it match that sequence, or I guess what I'm familiar with when you're using like really large molecules, those bands tell you sort of the molecular weight region that you're dealing with, in a sense. So, and again, I hope this wording is correct. If not, feel free to tell me to alter these slides. If the PI is greater than the pH of the buffer, the molecule will be overall positively charged and migrate towards the cathode, which is the negative anode. If the PI is less than the pH, the molecule will be negatively charged and migrate towards the anode, which is the positive electrode. So it's thin layer chromatography for large biomolecules. So the next couple slides are more of sort of philosophical in nature, not necessarily related to the science part of this. But it's all science, but more sort of thinking about things. As I've written these slides and developed this module over the years, like I said, I took a biochemistry class in graduate school and absolutely sucked at it just because it's like there's too much going on and I can't focus on like large things. But if you start asking these questions as humans, sentient beings do, where do we come from? Why do we exist? Is there anyone out there? Hopefully you can start seeing a connection of like all of the science classes you've taken. So there's this Operin-Haldane hypothesis, and it's based on abiogenesis. So A typically means like without, right? So the origin of life theory states that life has arisen from non-living matter, such as organic compounds. So again, I know that might be controversial to some, especially in today's political environment. Whether you're sort of on the creationist side or the evolution side. So evolution is basically, living things come from non-living things. And at a certain point, like not only do things live, but like we as humans have thought. And so can this bottle think? You know what I mean? Maybe over the course of the universe, it will evolve into a human that can think. But evolution is meant to encompass the environment. So our environment, Earth, that's the reaction vessel by which the synthesis of organic molecules, molecular self-replication, so that's what we'll talk about in terms of like DNA and RNA, self-assembly, autocatalysis, and the emergence of cell membranes. So that is a connection into all your biology courses. That's allowed for the transition of non-living matter to living entities. So from the atomic scale to the organismal scale to the human scale to thought. And I think what precipitated a lot of this was really the discovery of radioactivity and how sort of the offshoots of that allowed scientists to probe not only organic molecules, but things like cell cycles and metabolism and things like that. So this is a brief sort of prelude to the evolution of that, is the Miller-Urey experiment. And so Urey was a scientist, studied under Lewis, as in like the Lewis dot structures, eventually went to work for Bohr, and then started his independent career at Columbia. He eventually moved to the University of Chicago, but Urey was responsible for the isolation of deuterium. So that's the stable isotope of hydrogen. And so again, that relates back to humans as our understanding of the structure of the atom. So again, stable isotope, and then you have radioactive isotopes. So that's, this atom that is emitting radioactivity, which is like fascinating because it's always existed, right? But this nucleus in time, Becquerel and Curie, it's like, I noticed this weird thing, you know what I mean? And so that sparked really, I think, the world we live in today. I mean, you wouldn't have cell phones without knowledge of all of this stuff. So the University of Chicago was, a hot spot for radioactivity because of the work of Fermi and the Manhattan Project for World War II. Miller was a graduate student that went to Chicago to study nuclear physics, but eventually became interested in other things. And he had this idea of abiogenesis, like how can we start running experiments on this in the lab, which would be like, I don't know, nowadays if you thought of that, it'd be like there's so many variables and factors involved, I wouldn't even know where to start. But the experiment, this gets thrown around a lot. It's actually like a beautiful experiment because it's so, in a way, simple but very complex. So he designed an apparatus that contained the molecules of Earth's atmosphere, so methane, ammonia, hydrogen, and water. So the water, which is the ocean, and then hydrogen and methane and ammonia, sort of the atmosphere, was heated to 100 degrees, and then electrical discharges were applied to that to mimic lightning, so sort of weather. In that, to simulate a prebiotic atmosphere. So after a couple days, weeks, months, he was able to isolate from 1, 2, 3, those 4 different compounds, now isolate organic molecules. So glycine, alpha-alanine, beta-alanine, aspartic acid, and aminobutyric acid. So, in the 1940s, NMR was not a thing. And so he basically used thin layer chromatography of the mixture and references of those individual amino acids to say, okay, these things co-spot under these elution conditions. So it should be this. And here's just a pictorial diagram of what that looked like. So here's the ocean in the boiling water. So that gets your steam that's going up into the gases where the electricity is discharged. So that's providing the energy to, break bonds. You form these reactive intermediates that are colliding to form new molecules. They're floating around in there, and then it's condensed back in here. And down here is the trap where you can empty things out every couple days to see what's happening. So methane is the carbon source, ammonia the nitrogen source, water oxygen, and hydrogen is the hydrogen source. So in a way, it's Earth in a lab on a very rudimentary scale. And this is a picture from that paper that they published. And so normally you run a TLC plate from bottom to top, but if you look at the way this picture is, on the one axis, they ran butanol and acetic acid as the eluent. They took that plate and then flipped it 90 degrees and ran phenol as the eluent on the other axis. So that spot is aspartic acid, the big one is glycine. So that's the simplest one, right? So that makes sense of why it's there at so much concentration. Alpha-alanine, beta-alanine, alpha aminobutyric acid, and then I'm not sure what A and B are. But that's pretty neat in the sense of, knowing human knowledge and history up to that point. Like it represents a nice, beautiful experiment of, okay, these simple building blocks have made something more complex in a couple days. So the New York Times published an article on this, and I think I've linked all these things in Canvas if you want to read further, but these are the actual vials that contain those samples that were isolated from that. And people have gone back, they've isolated or reanalyzed these using modern instrumentation. And they've actually found that in his original experiments, like a lot more were produced. They just couldn't be like visualized under those conditions. And nowadays people do iterations of this that are more complex with fancier apparati, controlling the electricity, the concentrations of things. And I believe you can pretty much make any of the proteinogenic amino acids under these conditions. So after this was published, Richard Feynman, who also worked on the Manhattan Project, he won a Nobel Prize, I think, 1963 or 4 or something in physics for his description of the atom, wrote this poem. Feynman was, I think if you like scientists, he's probably one that you should read about. He was obviously extraordinarily intelligent and very creative as well. So not only with research, but he was also like played the bongos and did stuff within the art world. So he wrote this poem called "A Universe of Atoms, an Atom in the Universe," which you can read. But the, I guess, stanza that stands out the most. So stands at the sea, wonders at wondering, I, a universe of atoms, an atom in the universe. So from the atom to human to the cosmos, we're made from these basic building blocks. And the experiment on Earth, we exist, but does that exist elsewhere? And so, NASA has groups that do astrobiology. The James Webb Space Telescope, if you follow that, they'll look at like exoplanets, their spectroscopic signatures to see, okay, I have no idea how they do this, but they can say, yes, water exists on that planet 100 light years away. That's pretty wild to think about. And so the implications of that would then be, if there's water there, is there life there? So where do we come from and why do we exist? So for you to sit here today, there basically needs to be 10 generations before you. And so each generation requires 2, right? So your parents, and then your 2 to the n, is that, I think. So you eventually get up to overall 10 generations back from you. 2,046 people walk the earth so that you sit here today listening to me in this class, whether that's fate or whatever. It's pretty wild to think about. What we're going to do over the next 4 or 5 lectures, whatever it is sort of dial back down to the molecular structure. How do we make peptides from amino acids? How do they become proteins? What are the function of proteins? And then how does DNA get translated and transcribed into proteins? So the amino acids are an alphabet. DNA is an alphabet. What is the ultimate connection between these 2 alphabets? And that resulted in Francis and Crick. You've heard of them. Why did they win the Nobel Prize? So what was their recognition of the issue and the connection? So as you'll see, we're still going to learn some organic chemistry, but what are the implications of that in large biomolecules? And then anything downstream of that is the rest of your degree curriculum. So there is a brief quiz for today, pretty simple, based on the amino acids. I'll get the assignments set up in Canvas for this stuff, and then your Module 4 exam is on Wednesday. So